Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 24th November 2020. As all of us know, we are into the revision mode and we are revising an important chapter number 10, cell cycle and cell division. Why I said that this chapter is important is that this is not only as part of syllabus we need to study, but also as something that is going on every second in our body. Hence, as a biology student, we need to know what are all that goes on in our body so that we have the knowledge of what is going on inside us, not only outside us. In our previous session, we were talking about mitosis, the 4 phases of mitosis, important characteristics of each stage of mitosis, and now significance of mitosis also we have studied. And now we are going to go for next type of cell division. Normally we call it as RD, reduction division, that is meiosis. And meiosis is more important than mitosis because it forms the cells that are responsible for creation of all human beings, all animals and all the plants. That is, in case of animals including man, it is the sperm and the ovum, and we call it biologically gametes. So without gametes in sexual reproduction, there is no question of formation of zygote, and without zygote there is no question of life. Hence, thanks to meiosis, for meiosis creates sperm and ovum in case of animals including human being, and in case of plants it is pollen grain and the ovum, without which no life exists. So thanks to meiosis. Second, it also ensures that 46 chromosomes, 2 sets of chromosomes, 23 into 2, we call it 2n. So the 2n is getting reduced to n. That means diploid cell is becoming haploid. That is, the cells present in the testis, for example, testicular cell will have 46 chromosomes. When a testicular cell undergoes meiosis and forms a sperm, 46 chromosome number is getting reduced to 23. Similarly, when a ovarian cell having 46 chromosomes undergoes meiosis, the ovum that is formed will get 23. So, when both ovum and sperm mix together, sperm 23, ovum 23. 23 plus 23, 46. Zygote gets 46. That becomes a human being, human zygote. So thanks to meiosis again, as it helps us to maintain the species identity. So with that in our background, thanking the meiosis, let's move on to what exactly is the meiosis. So the production of offspring by sexual reproduction includes the fusion of 2 gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells by cell division that reduces the chromosome number to half, which results in the production of haploid daughter cells. Like I said, sperm and ovum in case of animals, haploid daughter cells. Pollen grain and ovum, haploid daughter cells in plants. So this kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores diploidicity or diploid phase, like 23 from sperm, 23 from ovum, zygote 46. So the original mother, father having 46 chromosomes and zygote also having 46 chromosomes, the originality is restored. Original number, diploid number is restored. Meiosis occurs during gametogenesis in both plants and animals and forms haploid gametes. The key features of meiosis are as follows. Meiosis involves 2 sequential cycles of nuclear and cell division called meiotic 1 and meiotic 2, but only single cycle of DNA replication. DNA replication only single, but phases as such cell division is meiotic 1 or meiosis 1 and meiosis 2. And meiosis 1 is the first division of meiosis, it is the actual RD, reduction division. Meiosis involves pairing of homologous chromosomes and recombination between them. Next, 4 haploid cells are formed at the end of meiotic 2. Whereas in the case of mitosis, at the end of mitosis only 2 daughter cells are formed. Here it is 4 daughter cells are formed. And meiotic events can be grouped under the following phases. As you are seeing, meiosis 1, meiosis 2. Under meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. And this is followed by meiotic 2 or meiosis 2, prophase 2, metaphase 2, anaphase 2 and telophase 2. And in turn, prophase 1 has got subphases or substages, and they are 5 in number. First one is leptotene, second zygotene, third pachytene, fourth diplotene, and fifth diakinesis. So meiosis 1, the heterotypic or reduction division is the first division of meiosis that starts after the interphase of the cell cycle. During this division, number of chromosomes are reduced to half. 2n becomes n. Like mitosis, it is also studied under 4 stages, but only thing we are giving numbers as prophase 1, metaphase 1, anaphase 1, telophase 1. And under prophase 1, prophase 1 of the first meiotic division is typically longer and more complex when compared to other prophases of mitosis or meiosis. It is further divided into following 5 subphases. So first one, we talk about the leptotene. During leptotene, the compaction, the chromosomes becomes distinct and visible under microscope. Compaction of chromosomes continue throughout the leptotene phase. So here you can see the formation of nucleus, nucleolus will be there, centrioles will be there. Chromosomes only will be becoming short and thick. That is the characteristics of leptotene. And during zygotene, the chromosomes start pairing together. So synapses will start. Homologous chromosomes will be formed. The paired chromosomes which look like are called homologous chromosomes. And the synaptonymal complex formed by pair of homologous chromosomes is called bivalent or tetrad. So here pairing of chromosomes starts basically in zygotene, and this stage is characterized by formation of synapses. That is, chromosomes start pairing together, and this formation of pairs of chromosomes, homologous chromosomes, is called synapses. And the homologous chromosomes forms pairs. We call it bivalent or sometimes the 4 tetrads. However, they are more closely visible in pachytene. The third stage, pachytene, crossing over between non-sister chromatids of homologous chromosomes occurs for exchange of genetic materials. The most important stage in the entire meiosis is pachytene because crossing over takes place. And crossing over means chromatid of one chromosome crosses over to chromatid of another chromosome. That's why we are saying non-sister chromatids, not sister chromatids. And because of this crossing over, when they come back to their original state, there is exchange of portions of non-sister chromatids. As a result, when portions of non-sister chromatids get exchanged, automatically the genes also get exchanged. As a result, exchange of genetic material takes place, and exchange of genetic materials lead to formation of varieties. So for the formation of varieties, pachytene is the most important stage. This crossing over is enzyme mediated process which involves an enzyme called recombinase. Next, diplotene. And in diplotene, it is recognized by dissolution of synaptonymal complex and tendency to separation of bivalent except at the site of crossing over. This forms X-like structure called chiasmata. That means in diplotene you can see the formation of hybrid chromosomes, and the hybrid chromosomes having chiasma. That means they cross each other, the point of overlapping of non-sister chromatids, that point of overlapping or crossing over is called chiasma. So many chiasma you can see, formation of many chiasma in the diplotin. So it is recognized by the dissolution of the synaptonymal complex, and the X-shaped structures called chiasmata are very clearly formed. In oocytes of some vertebrates, diplotin can last for months or even years. So that is the recognition of diplotin. And diakinesis, it is marked by terminalization of chiasmata. The nuclear membrane breaks and nucleolus disappears. So diakinesis is the final stage of prophase 1. This is marked by terminalization of chiasmata. So the chromosomes are coming back, and formation of hybrid chromosomes is the resultant of this. During this phase, the chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears and nuclear envelope also breaks down. Diakinesis represents transition of metaphase 1. That means prophase 1 gets completed once diakinesis is completed. So these are all very important. Prophase 1 is the most important phase because of exchange of genetic material. Thanks to crossing over, thanks to chiasma, and the thanks to also the formation of the crossing over and hybrid chromosomes. We'll continue in our next session about metaphase 1. Until then, keep on practicing diagrams, label the diagrams, and understand the significance of each label that you label in the diagram. So I am expecting you to post 5 diagrams of prophase 1, that is leptotin, zygotin, pachytin, diplotin, diakinesis with labeling.